Okay, so I'm gonna share my screen and let's check the homeworks that we were talking about, and maybe it's gonna be a recap for you and you can ask your questions. So first session homework was about creating this tree structure. Here we copied some of the files. We created links for some of the files here. And but how we did, and we created the directories. So let me do that for you. For example, I'm gonna go to my do I have that directory? No. Okay, let me see how much space I have. First, I'm checking how much space I have. See, I have only 312 megabytes. So maybe if I do all these things, it's gonna take more. No, it's not gonna take more space, but I'm not gonna use here because I will use for other things. So I'm gonna go to my project space. I'm changing my directory. And I'm checking how much space I have here. So I have 457 gigabytes free space. So this df-h dot command told me that, and I'll check if I have RNA-seq directory. No, I don't. So I'm just gonna create this directory, RNA-seq. So to organize yourself in the project space or nearline space, because you're gonna share this places with other people in your lab, what you can do, you can create another directory for yourself if you are using, this is my username, for example, or you can use like email username if you want. And the people know that this directories belong to you by just looking the directory name. And I will do it here, and I will create my RNA-seq directory here. And I'll show where I am now. I'm here, and I'm gonna go in here. I changed the directory. Okay, so now I'm ready to create these three directories, mm10, procs, and reads. So how you can do this? Just with one command, actually. So what was it? mm10, procs, and reads. See, you can do it like this. This will create all these three directories at the same time. See, I have all of it now. When I do tree, I have 3 of these folders and they're empty now. So first I'm going to go to this first folder. And I want to put my genome files here about MM10. Just this is for organization, all these things we are doing now. So in this case, usually I don't copy these big files. In this case, this is not big, but usually FASTA files like whole genomes for human mouse, they're pretty big. So I'm gonna create a link for this using this command, and you can just use dot here, means I'm gonna create it here on this directory. Or you can just write the name of the link you want to create like this. It's up to you how you want to create. If you just use dot, it's gonna use this name as a link name. See, I have it now. When I do this, see, it's showing that specific file. And now I have this ucc.dtf file, and it's on this directory, and I want to copy it here. So you're going to use cp command. This is the source, and you can use dot again just to show here. Or if you want to change the name or use the same name, you can do like this too. It's up to you. I'm just going to do that again. Okay. So the file is here. This is not here. By just looking ls-l will tell you which ones are physically there in that directory. And then this is done now. Just to check if it is done or not, you can do the tree. Oops. See, this is how I want. Look, this is how I want. Now I'm going to go to this directory, procs. And in this case, this is different. It's on that file is on a URL on the internet somewhere. It could be anywhere. So what you can do, you can go to that directory. It's going to be a program, and you can use wget command to get that file. Not this one. So I copied. I'm just going to copy, and paste. See, I got this file here now. See, whole file came. And if you want to look what it is inside, I'm just going to do head. See, this is the program, or tail. So, or you can do more just to look at the whole program. This is a Perl program. And we have reads. I'm going to go to the reads. And I'm there now. And the files are here. So you can do lots of things here to copy these files. There could be more files there. The suffix of this files could be different. You could only copy the files that have the FQ suffix here, like this. File type is FASTQ, but you can just use the star.fq, or you could do star in this case. But there could be many other files there. That's why it's good to learn like this. And I'm just going to copy these FASTQ files to here. So everything is going to come here, and they are here now. See? And now when I do this tree, so everything is like how I want. And I'm going to do tree here. See, this was exactly like this tree. And now I want to create a tar file, tar. So when you write like this, xvfz, and when you do tar.gzip, for example, this is the name of the tar file, and I'm gonna put all this directory. So this is the name of the file. These are the parameters to, sorry. X is for extraction. C is going to be for like putting them into a tar file, and Z there is going to be to zip, and VNF I usually use this variables and foresight. That's just putting everything in. And you could put other directories there too on the side, like, or files, directories, links, whatever you want into, like, you could write here, but I organize like this into one directory and putting like this. So, and now it is doing this, and then I'm gonna just look. So this should be the size of the directory that after you do everything right, this could be the size. So some of you, I saw that you didn't zip, you just did something without zipping. I'm going to show you that version without Z command. Oops. It's going to be larger. See, I saw some of you guys did like that. I think this is that homework. Any questions about here? I think it was easy. You should all be able to do this. Okay, this is something you're going to do a lot. Even you use Dolphin Next, you need to organize your directory structure yourself. Sometimes you get your backups yourself. These things are going to be important. So please learn to organize your own directories. And later you're going to learn, like after you zip, and you need to remove some files to create more space. Okay, I removed, for example, using rm command, and I'm going to remove all that RNA-seq folder. See, it's gone. Now I have space, like all this space is free now that I can use for other things. If I need back all these files, this could be my backup. And
excuse me, Alpa, we couldn't see your screen.
Yeah, you're right. Thank you. So you can delete what I did was that I just removed these files, this one and this one, and then you can extract the files like this. See, they are back now. Free RNA-seq. They are back now. So you can easily delete the files, but make sure while you delete, you get copies into at least 2 different places. One of them could be somewhere I don't know here if you're going to use it, or you can use your some external drive, but those are dangerous. So be careful to get like your backups somewhere. Like I do usually, if I'll use them, I put them on Amazon, and I put Amazon not that expensive, so you can talk with your PI and always back up your, especially your FASTQ files, like initial raw files to Amazon. That that's gonna save you a lot of time if you can do that. Okay, so I'm gonna remove this again. Oops. RM is not going to work because it's a directory. I need to use this RM minus RF to remove that directory, and it's gone. Okay, good. Now I'm gonna go to the second week, second session. Is that this bootcamp? Session 2. We had a homework here. So in our cluster, how many jobs are running in the long and short interactive queues? So we had a command called bqueues. Was it like this? Queue, something like this. Yeah. bqueues are going to tell whatever running in our cluster. So we're gonna focus on short and long queues. So in the short queue, these many jobs exist, submitted, and these many jobs are pending, 98, and 399 jobs are running now. In our long queue, 6,000, around 6,000 ones are running here. These many, 3,000 pending, 10,000 jobs submitted. So see, this looks a little busier, but this is short is less busier. So what we do, for example, if you need to finish something fast, sometimes you try to submit them to short queue, like many of the jobs, and only the ones that requires long queue, you submit them there. So don't use long queue all the time, because many jobs can finish in the short queue. Sometimes even when it is long, I split the file into smaller chunks and then map them like that if I need them, and then merge them using SumTools merge function, like if I need it fast. So you can think like that too. You don't need to always submit long queue. Even the job might take longer, you can split it, the files, with like those commands, like you can use like head minus n 1 million of the file, and maybe I can write it here, head like minus n 1 million. There are other split functions, but basically you can do something like this. The file name, file.fq to file first.fq, for example. If you want to do these kind of things manually, it's going to take a lot of time, but you can split the files and do each million separately, and then submit them short queue, and then get the results and then merge them. So this is, and the second one is how many jobs are pending. We did that, run a sleep job for 100 seconds and kill it. So I'm just gonna do like this, the B sub. You can define all the other things, but I'm gonna just submit it like this, and my job will be like sleep 100. So I submit it, and with B jobs, I can, it's coming, it's here now. So it's gonna sleep 100 seconds, but if you want to kill this job, you can just do B kill, and it's done, it's gone. You can look at that job again with B jobs minus A. See, it's here. And the last one, I think in the homework. Okay. So you can look at that, by the way, what happened with B jobs minus L2. So in this case, see, I killed it, job killed by owner. So this could be anything else. You have to track down every single job you submit into the cluster. Okay, if they fail without your knowledge, maybe there could be some files there that you might think that it's finished, but it's not. You have to actually get the right exit here. It has to complete it without any error. So now I'll do another job that I will just put any command, like, when you run this AA, for example, here, that command doesn't exist. So it's going to give an error. So I'm going to submit this command with B jobs. You can actually submit them with the things you learn here with all these, like memory, time, queue, everything like this. Let's do that like this now. To short queue, I'm just going to add minus, like, this command. That command doesn't exist, but I want to put the output to an output.txt file. Where did I put that? Logs. So you can put the output like this or error into another file, like error file. So I'm just gonna copy them. You can put anywhere, by the way, but make sure you're not putting in between these parameters. So for example, minus and 1 should be together. So you cannot put other parameters in between, okay? But the order of minus n, 1, minus r usage, minus w, 20, minus q short can be in different order, okay? So for example, I'm gonna put them here. But I'll put the error into this folder that I'm working on now, and the output will be there too. This is gonna be 0, and I'm supposed to see the error here in this file, okay? Oops. What did I do wrong? Oh, sorry. It's not B jobs. Your command is B sub. Okay. Now I submit it. And then I will see it's finished. And how I can see it's here. See, this was my job ID. This was job name. And it was in the short queue. So let's see. Interesting. It should here. Okay. See, output an error. It took time. That they should be here, by the way. This was exit and I did ls, but they weren't here. So our storage is sometimes slow. It's writing in the background, but you need to wait a couple of seconds when you do these kind of operations in the cluster. So oops, I'm gonna do look at this. See, I see the same thing here. So you can track down your jobs with this error and output. Okay. Any questions about this homework? So if you can do all these things, you're ready to use cluster. The only thing you need to remember here was like don't use. Okay. So don't use the head node for the big operations. Just get a like an interactive node. So what I do, I put like one of the interactive node script into this file in my bin directory. See, B sub interactive 120 minutes, and as I don't want to use SGI, that's why I put such things. But you can just put this command here, interactive node, this command into a file every time when you want to run, and then change the permissions of that file to 775. As you can see, this is executable. Every time when I do like Q login, like I put it here. See, I don't need to remember how I was doing. I just do Q login. So you can do any file, by the way. It doesn't have to be Q login. But I'm now on this one of the nodes, and here, see, this is the node name. Then I can start using cluster. So this is what we want you to do. In the future, when you come to the cluster, if you want to start anything, just come get your node and then start running things there. Good. You can interrupt me anytime. So here, let's look at that some commands that we learned in the session 3. The homework was like submit 2 dummy jobs in the long queue and 3 dummy jobs to short queue, then get a list of your jobs that have been submitted to long queue only. So yeah, let's do real fast, like B jobs. Sorry, not B jobs, B sub. So sleep 300. I'm going to submit this to long queue minus Q 300. Oh, sorry, minus Q long. My brain is not here today. So another one I did, and then I'm going to submit 3 to the short, short, another short. And I have 5 jobs. See, this is all. And I have 1 interactive that I'm on, and the others are there. So just to get the ones on long, you can just do this grep long. See, the grep is for that. Like when you have larger files, if you just want to focus on something using a keyword, grep is your command. I use that a lot. Like the other day, for example, I did a quantification of some wild type sequence in a pipeline, and that person said that, oh no, this cannot be true. You know, this cannot be true. This wild type sequence shouldn't be that many in the FASTQ files. And what I did was I just put that wild type sequence like AC, GT, AC, whatever, wild type, and then put the FASTQ file, like, you know, whatever file that FASTQ, for example, and then did this and divide it by 4. And I showed like this. You can do it like alc print $0 divided by 4. I did this and showed that this was the case, and she believed in me. Otherwise, she wouldn't. Okay, so this is like sometimes these kind of comments are going to help you to do your own checks. Okay, that's why I learn. But usually in that case, I was using exact match. But in the future, you're gonna allow some mismatches while mapping. But still, you know, even you allow mismatches, those mismatches will be only 1% or 2% of all that sequence exists in your FASTQ files. So that's why when you grep, you should get a huge amount of those reads in your files. So you can try that. Like, for example, you can get one of those reads from FGF21 and search for that gene that you worked on last week. So, and then you can grab one of those reads and see how many times you have those. So you have to have like good amount of those reads, even you do exact match. Okay. So this was the first, and the second one, let's do this one. So what we want to do is like when you just grab, for example, this gene FGF21, in this case, in this homework, what we were doing, in this file, we wanted to get this line in this format. You know, I'm changing the format because the format was like this. See, I just get the FGF21. This is a huge file, but I just get FGF21 gene, and these are the exons. This is the annotation of that gene. This is the start position of this exon. This is the end position of that exon. This is the strand of that gene. It's in the crick strand. Here, just the start codon starts here, stop codon starts here. And what I want to do is like, you can count like how many columns you want to get this FGF21 while you print that specific column. So what you do first, and also in this case, we want start codon. Start codon is defined like this in the third column, but I'm just going to do this first. Grep start codon. Oops. Sorry, that copy. So I'm just going to copy this. See, I get the start codon now. Now I'm going to reorder all the columns. I can use alg to reordering the columns. I'm using these kind of things a lot. That's why we are showing. So for example, we're going to put chromosome 7 first. And this is in the first column. And then I put tab in between. Some people are using like comma, you can, but you don't need to. And the second one, this is in the column 10, FGF21. You can count like 1, 2, 3, 10. And this is the start codon in the column 3. And this was the start position, column 4. So when you do that, I have this exactly like how I want it. So when you do this, so what I did was in the first command, I put the file here. Make sure you put the file here. I got questions like, they use this file at the end. It's not going to work. What we are doing basically, these bit pipes, what we are doing, the first command is going to bring us all this list. And the output is going to be generated. And the output could be sent to the screen like this, to the terminal, without like adding pipe or nothing. Or you can use pipe to send the output of that specific command. It could be anything, by the way. But in this case, we just get the FGF21 gene from that file. And then we put the output into another grep command, okay? And then we put it here. So grep is pretty nice. Like, you can even like, for example, if you want to color the start codon, if it is a huge file, you can say again, like another grep. It's maybe meaningless, but I'm just showing color always, for example. See, you can just emphasize some of the things if the file is like huge, or if you just want to see or emphasize something in that grep. So you can use this. It's very cool functionality. Okay, I think any questions about this grep and alg or VI? Again, like if you learn first 2 weeks, on top of that, if you learn these things, you're done. You're half bioinformatician. If you learn them enough, you can do many things with these things. Now, let's go back to the forms last week. I'm not going to go in detail. I'll give you more time about that session, but I will remind you maybe a couple of things here. So what we learned here, basically, we learned like how to load the modules and how to, let me go over a little bit just to explain. I got a couple of questions about it. So what we did, basically, we load the modules first. So what I see, like some people didn't load the module and try to run the command. So it's not going to work. So you need to load this module, for example, to run star or RSAM or something. And you have the versions here too. These are important. Just if you're doing these things manually, make sure to note which versions of the software you used, because they always change. And a year later, when you come back, if you want to create exactly the same run to compare your old data with the new data, for example, if you don't know that information, then you have to run everything again, So don't do that. Just write them down into a README file. I usually create README files to organize my runs. And usually I put all commands that I run for a specific project. If I'm doing these things manually, I put them into files, like the script files with VI, and then write some explanations about this. For example, what I do, let me show you like how I'm doing. Then you can learn doing that. For example, I'm here and I create like README file for myself, just to remind myself like what I'm doing here. Like if you put, you can say create directory. And then if you want to run them actually and execute them, you can even create like this. These are the commands you are running. So create directory, run, for example, run start command. And so first I load, for example, these guys, but don't put the dollars there because dollars is just for this tutorial, okay? Dollar is showing the command prompt that you need to run them. But in this case, if you want to run them, you need to put them like without dollars. Okay, now see, you can say here, load the modules. So, and then this will remind you later when you come to this directory, what you did here. You can look at your README file and see, oh, okay, I load these modules. I run this command with these parameters. Everything is organized. And I use this UCSC GTF file in that specific directory. For example, you have everything. And then you can do this. Next time when you do, it's going to be easier. Okay, let's go here. So this IGV, I'm just going to show, I'm not going to go over like everything there. You need to do it yourself. But here, your homework was about IGV. So let me check if I have an IGV here. Yes. So I'm going to give a couple of tricks here about IGV. Then, so IGV is a tool like, see, I have mouse here. So you can choose mouse, MM10. So the first thing you need to do here is you have to be sure which genome you are working on, okay? And if you didn't I skipped homework 3? Okay, sorry, I'll go back there. I skipped. So let me show this IGV first. I'm going to go back. So MM10, we use MM10 in this case, but it could be MM9 or HG19, HG18, whatever. So you have to choose the right genome, or you can actually load the genome here too. If you have your own genome, for example, if you use flu genome or COVID-19 genome, you just want to visualize your reads on that, you can download the file using FTP or put a web accessible area somewhere and then use the URL while loading the file. Okay, when you load, it's going to come. See, I did, for example, EGFP gene. Just for one gene, I wanted to see the distribution of the reads on that and how it worked. So you can, sorry, you can, because there are some problems sometimes, then you map something there and it doesn't work how it's supposed to work. That's why you need to check those things by yourself, your own eyes. That IGV is very good for that. And also you can use UCSC Genome Browser. There's an upload there too. So what I'm going to do now is I put this MM10 into somewhere web accessible. Put it somewhere here. Okay, so I have this MM10 FA, for example. You can, if you want to create your own genome, you can do this. You can right click here, copy link address, and then create your own genome. This is like smaller MM10, but it's fine because this is, I'm going to use this original version here. You can choose it MM10 too if you want to try now. But we created all these alignment files. You know, after you run all those steps in session 4, you're going to have this sorted and indexed alignment files. To be able to visualize your files, these files has to be sorted and indexed, okay? And how you do is in the tutorial, basically you need to use the sum tool sort function and put the inputs, it's there, and then index function, it's gonna create the index, but you're gonna write the file name here, the BAM file, and the output name. And then after you create them, you can change the name. I don't know how you wanna organize, but in the homework, what we wanted to do was like, for example, I want to load this. By the way, you can copy them into your computer too, or if they are on a URL like this, by the way, we can give the service to you too in the future. We can like create a web space for you, and then you can put some of the files there to visualize. So talk with us. If you need this kind of web accessible area, we can give a link with your user ID and the files you want to use, like you can put them, visualize them, and then delete them later, for example. So what I'm going to do now, I'm going to copy this link. And in IGV, I will load from URL. Here, there's file, load from URL, and I will put it here. And then if there's no index file, it's going to give an error. But there is here. So this was RSAM file, and I want to show you the difference between RSAM and STAR. And one of them was transcriptomic alignment. The other one was genomic plus transcriptomic alignment. So in the second one, we're gonna see some reads. I don't know why this is doing that, but it doesn't allow me to write 21. I don't know, there's a bug, I guess. Okay, so I came here. See, you can see the difference here. So one of them was here, control 1, RSAM. This is STAR. As you can see, you don't see any reads in between in this internal intronic regions here. See, in this part, in this track, in the last track, you see whole genome. In the GTF file, by the way, you can load your GTF file yourself too here. If you have your own GTF file, if you change something, or if you annotate a new gene, you can actually use them. For that GTF file you use, and you saw the coordinates. See, these coordinates are here. Whatever number you see there is here actually. So this is the start and end position because it's in the other strand, in the quick strand. So these are start codon, stop codon here, and you see 5' and 3' UTRs and intronic regions. So in the intronic regions, there are some reads here in STAR. So this is important because sometimes if you have a lot of DNA contamination, you can see a lot of reads everywhere else. You know, you can see how much DNA contamination you have, or these are maybe unspliced versions of this gene. You're going to see a little of these, but not much. Okay, so these are like sorted them. These tracks are the exact reads. So you see reads themselves. You can zoom in and zoom out here. See, these are reads themselves. So when you click, you can see the name of the read here, see? And all the information about that read. So, but if you just want to focus on the coverage, so this is the accumulation of all those reads in those positions, okay? If you just want to focus on there, you can come here. Usually I do like that. I remove these. I select them with shift and multiple. You can select multiple commands and then you can remove them. See, I have STAR and like only these two. You can change their colors. For example, I'm gonna change the color to I don't know, green. So you can change the color like this. And what I'm gonna do, I'm gonna load one more. Like for example, this one, experiments, just to show you load from URL. And I'll remove these. And I'll make this, for example, red. See, you can add as many tracks as you want and visualize. See, there are little reads here, not much. So this is fine. So you do these checks. You go some gene state or some positions in the genome by changing or searching here. You can do those things. And one more thing you could do, you can look multiple genes by defining your gene list here. For example, I have my own list here created before, but basically you just press this new button and edit that or add all the genes you want to look and then close. And then when you say view, so it's going to split the screen and you can look all these genes like this. So, but when you look like this, it's normalizing or using like one, the one of first one, for example, in this case, this is the largest. The others are looking like much lower here. So this is like just to compare different genes. You know, you can use this thing. This is not here. Why? Because the gene is, as you can see here, it's too big. that you need to come here and, oops. Oh, where was that? Why can't I? Yeah, you need to basically, it's here. You need to zoom in here to be able to see the reads there, if there are, okay? Basically, I'm zooming in now, but this is the largest, I guess, yeah. But there's nothing here, that's why we don't see anything. But basically, you know, it's working like this. If you want to change the height, or you want to put in like these tracks into log scale, you know, those are here. You can do group auto scale, or you can do only for the groups, for example, only the controls. You can separate this one from the others or something, like, you know, you might have different type of tracks. In this case, I only have RNA-seq. I should use them all in the same group. But if you have chip data, if you have attack data, if you want to compare, you might need to group them in differently, you know, because their peak heights will be different. So all these things are here. I want you to play all these things here. And also there is preferences in the view that you can, I always set this, for example, normalized coverage data. So this is normalizing the coverage data. It's good to compare different RNA-seq tracks here if you normalize. Otherwise, you know, if you have 1 million read in one, 10 million in the other, they're not gonna look like similar. That's why you need to normalize them. So yeah, there are many things, colors, and you can play and learn more. There are other tutorials on the internet too. Okay, so last thing, let's go back to, where was it? Session 3. What was session 3? I forgot that. Okay, for this is, let's start from the beginning. Session 3, genomic resources. Okay, here. So do we have homework here? No. Okay. Do we have homework? No. And fast Q, we had homework here. Okay, so what is the nucleotide sequence for this specific file? So what we are going to do basically is this. So again, we are going to use grep function, and we need to find this specific read. Sometimes, you know, you get an error in our pipelines too. It gives you this read name. It's going to tell you that, hey, there's a problem in this read. What you need to do, you need to look at that read. What's wrong with that read? And in this case, I don't have those files here, but I am glad I get a backup. And I will just open my backup. And I'll go to that file. So the file was this one. So basically, I'm just going to grab this one. But when I just grab like this, oh, it's not there. Is it in the second one? Sorry, replica 2. Okay. See, I get this, but I cannot see the read here. I could only grab one thing, but I know that read should be in the next line. So if you want to print one line more, like after that, after it finds this read name, you can use this, you know, A minus A1. It's gonna write the read. If you want to do like 3 more lines to get whole read with the quality information too, you can do this with 3. So this is whole read. If you want to see the previous 4 lines, previous read, you can say minus before, I guess, after before. It's B minus B4. So this was the previous read. This is the read that I grabbed. I did the grab. So I got like where I am now. If there's a problem here in this read, you can assess it. Sometimes what's happening, like, you know, you lose like one nucleotide here and it gives error sometimes because of a reason, you know. So this is, excuse me, this is how you can actually see this. Is this that read, by the way? Let me see if I'm doing right or no, not. I'm just gonna grab this. This is my read. I'm gonna grab my read. This read. Yes, this is what the right read, that it worked. See, if I want to do this, that's why it's cool. Like I want to print the previous read again after I find this, minus B1. It's gonna be the previous one. And I'm gonna do 2 more lines after 2. It's gonna be the same thing. Minus 2, or minus A, 2. So it's the same thing, but I wanna color this, color whatever I find. That's why color is cool. See, this is the read name and the grep is here. But if I would do this at the beginning here, it was going to color only my read name. Okay. So yeah, these are cool functionalities you can play. And other homework. Okay, so this is advanced homework. So what we're gonna do is basically we need to convert a we need to read it from a SAM file. No, using a single command line, print the last read in the BAM file. Okay, it's the BAM file. We need to go to the BAM file and convert it to FASTQ ourselves. So we know where they are, like where the read name is in the which column and the read itself. And we're going to put a plus sign and we're going to put the alignment file, this quality information. So I need to find a BAM file. I'm just going to find something like, for example, where I had maybe so I'm gonna go to that location and the biocore. Oh, I don't remember where I put those BAM files. That's why. But I need, for example, okay, so I have some BAM files here, those BAM files. So what I'm going to do, I'm going to first load the module, some tools. For example, I'm going to load this version. And I'm going to do some tools view and this BAM file. And I only need the last one, tail. So these are last 10 lines, but I need only one. Tail minus 1. Let me run this. So, okay. So the read name is here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 is my read. 11 is going to be my quality. So basically, you're going to just do this out. Print $1. And I'm going to put the enter character $10. I'm going to put the plus. Sorry, not now. Now, plus here. See, I just put this. Enter character plus, and then another enter character and level. See, done. So you can do like this. It's kind of cool. I'm gonna do maybe 2. See, I get like last 2 reads. So sometimes, you know, there are converters too. You can convert them to FASTQ. You can download a file like that, but you can do yourself too with one line. See, it's not that difficult. So we do these kind of things a lot, like converting some BEM files to BEM files, BEM files to FASTQ files, FASTQ align them again. So basically, for example, when you can do this is, for example, you just want to get only reads mapped to some specific region. So you have some tools for that. And some tools view. And there was like this, what is it? I forgot. Whatever, I forgot, I can tell you later, but you can get like specific region, for example, only chromosome 7, starting from 1 million, look, and 2 million. So you can get only those reads. Sometimes you need that. And then you want to use those reads and align to some genes to yourself. You just get a subset using this. I think it was -L or something. I don't remember exactly, but there are ways to do it. If anybody wants to do such things, you can always ask us. And then we look usually these kind of things to internet too. We search like how to do these things to remember. I did those things a lot, but these days, I guess, I'm not working on alignment files a lot. Okay, I think we did the whole homework, and I think let's take a break now, and we will come back maybe in 10 minutes. I'll be here to answer your questions, and we're gonna start my favorite part, Dolphin Next, in 10 minutes. Okay, 10:05. Okay, let's start. So, okay, now this is my favorite section, and I'll talk about our D suite, Dolphin Next, and D Meta, Dolphin Meta, and Dolphin Portal. Like, let me share my screen again. And thanks to Arthur, he created amazing slides to tell the story. So I'm just gonna use them, and I added a couple of stuff. So what I'm gonna do today, so I'll give this example, our Vitiligo project that we are working on to tell how we are using D Suite and why we needed D Suite or Dolphin Next or the other applications that we created. So this is this project is done in John Harris lab, and Manuel, they are doing this collaboration. And in our lab, mostly Arthur is working on this project. So, but this is an example. So basically what we are doing in this project, that the people in the hospital are collecting some samples from the patients, some donors, doing this blistering method. And then they are sending the sequencing facility to get the single cell sequencing reads and results. And then they send the results to Arthur, and then Arthur is doing all these analysis. Basically, what we want to do here is we want to centralize all this data management, because in this project we had more than 170 patients, and their data was coming like in time, and some of the patients visited the hospital a couple of times, that they collected samples in a couple of different time points, that after the treatment, before the treatment, and they have different diseases too, that we need to keep track all that information in somewhere in a database, because before they tried this with Excel sheets, that it didn't work, and it caused a couple of problems. For example, one patient visited the hospital 2 times, and they gave new patient ID to that person. And then, they were thinking that this is coming from different patient, but after the analysis, they realized that that data is very similar to each other, and they realized that this is the same patient. But after bioinformatics analysis, actually, they realized this reality, and then they go back and looked at the lab notes, and then they realized that this is the same person. So we need to centralize all this effort. That's why I'm going to talk about our aims, why we created this. And later, when you do these things, and the people start working on different things, like, for example, some band scientists started doing RNA-seq experiments, and others were using the same donor samples, biosamples, and derive new samples from them to perform some ATAC-seq, single-cell ATAC-seq experiments or ChIP-seq experiments on the same data, same sample. So we needed this data aggregation. And sometimes they did like bulk RNA-seq, and they wanted to use single-cell RNA-seq together with bulk RNA-seq, just to profile the percentage of that data. In this disease, as you can see here, like there are some white patches, and those, so when they were blistering this, what they did was like they collected samples from those white patches versus the healthy region. So they were calling this as like lesional and non-lesional regions. And also they get some, like they did collections from the border of that region. It's called like perilesional data that they collected. So it looks like this on the pictures. But basically what we want to do, we just want to collect the sample and start collecting all that information coming from that specific project. So think about like this for your own projects too in the future. Even you have 20 samples. If you start like this, it will make your job much easier. Talk with us, we can create your project, and you can start collecting all that information about your samples according to your project in our suite. And then in the future, even before you send them to sequencing, by the way, you can start entering all that information method before you start doing experiments. You can organize, design, and then enter that information to the system. When the files come back, you can enter them later, and then the data will be processed in Dolphin Next, and you're gonna get the results and do other downstream analysis. After you learn all these analysis techniques, you can use the things that we are showing here, or when you have the all information collected in a single space, then you can use them in other software too. You can download and upload them to other software that exist, in the market. If you have, for example, Ingenuity or I don't know, DNA Nexus, 7Verges. So there are many other companies doing these kind of things too. There are some free software too, like Galaxy. So whatever you like, but the only thing you need to do is you need to start from the beginning to collect that information that we wanted to do. At the end, what you're going to get in this case, for example, you're going to get this kind of t-SNP plots coming from that patient, or we are using, in this case, for example, 170 patient data in a single t-SNP plot here, and characterize all the cell types coming from there. And then we can do answer lots of biological questions when you have the metadata attached with your process data. This is the real aim. We just want to create this kind of one unified environment, okay? Because as you can tell here on this side, when you only focus on this side, like pre-processing, there are lots of things you're doing there, but nobody knows what you're doing on the hospital side, or if you're a bioinformatician, they focus on this alignment quantification and data analysis, that this is the things that we are doing here. But when you are a bench scientist, if you want to communicate with a bioinformatician, if you are not working in a single unified environment, then there, you're going to see that there will be some gaps when you are communicating with them, unless you put all that information in a centralized place. So that's, and when we look at the other side of the story, so usually, when you are performing any methodology, any experiment, any protocol, you're using existing or published methods, unless you're creating your own. But the thing is, when you want to replicate their results, usually it's so hard, because they create this, even a year ago. I will give an example. A year ago, a paper, and I wanted to create their results, and took a month just to reproduce their results, because even they shared all the source code in an open source GitHub repository. This is GitHub, by the way. These symbols are GitHub, and they are using all engineering to share the results usually, but I couldn't replicate because they didn't share the containers, Docker and Singularity. So the containers are the systems that will allow you to install all these things in this, under this sea level, okay? So it is creating a machine like you have now. You are using your laptop, for example. It's getting a snapshot of that machine, and whatever you installed, whatever programs you installed on that machine, and their versions are going to be in these containers, okay? So if the systems or any paper are not sharing their Docker images in the future, it's going to be really hard to replicate their results, because all the programs, operating systems, all the parameters they use while they're running the software there, or the reference files, or sometimes some software are going to be outdated, and you will no longer install that software into that specific operating system that you have. So these are huge bottlenecks for reproducible science. So that's why you're going to see that we created a system that is using Nextflow and all this containerization, open source, and you can see all the code on GitHub. And we do all the tests with Travis. This T here, that Travis is a tool that are helping us to test all the things we do, and it creates less errors in the long run that reduce our time we spend to create all these pipelines using Nextflow. And then the results, whatever we create, you're going to see that they can go to our studio that we're going to learn today. Hopefully we're going to start learning, and Jupyter. So what we basically do, we create pipelines, all these tools, and use RStudio and Jupyter to do like some more downstream analysis. That's why I suggest you don't need to focus on too much about this part yet, unless you want to be a bioinformatician who is following all the standards to create like a reproducible pipeline for your analysis. We can focus on here. This is our job. Okay, we are working on this part, but you can focus mostly on RStudio and Jupyter part. That after you process, still you're going to use all the software here using our interfaces in Dolphin Next, but you can focus on RStudio and Jupyter to learn and to tweak the data and to create your own plots and play with your data. This is the paper that I was talking about. So this was about single-cell RNA-seq data, and this is Rachel Green's paper from her lab, and they created a beautiful paper, and they used the latest technologies that time. And but you know these versions are here, so we know like what versions there. But imagine like there are this. I just put a couple of them here, but there are 100 more like dependencies here, and installing them, having the same versions are almost impossible. Okay, and that's why you cannot recreate what they did. Like you cannot recreate this beautiful thing that all these t-SNP plots or PCA plots here in this case yourself. So, but what, like, you know, a person came from a lab to me, and he said that, you know, I love this paper. Can I color my own genes on these plots? And I said that, yeah, you can. I started like this. I'm always optimistic. And I created like similar plots because I couldn't find and install all the same versions. Everything exists here because it's almost impossible, because the R version that time was 3.3.3 and the version I was using was 4, for example. And it took like a month actually to create exactly the same thing. And I realized that, not that time, but before I realized, of course, but everybody needs this, you know, like you see a beautiful paper, you just want to color your own genes on those plots, for example. And this is ongoing problem. But lots of people started thinking how we can create reproducible pipelines. Even they share the code with us like this. But what I saw in their code was they usually use this exact paths of the software or the data that they had. This is a huge problem. So when you're writing your code, make sure just put them into variables or put them outside as a parameter in the future when you're writing your code, and make other people's in the future their job easier. Okay, so because when they do like this, and I was looking, I don't have this directory, this is his cluster, his directories, and I don't have those files, and I had to write them, hey, I don't have these files, could you please share with me that I want to reproduce your results? And they never respond. So this is sometimes, you know, it happens, you know, some people are coming and going, they are not working in that like, you know, lab. And then I sent to Rachel Green helped me, and then I could reproduce the results. But what I'm saying is, like, sometimes nobody can even answer you. So when you create your own pipelines in the future, if you create pipelines like how we create today, then nobody's going to need you, and they can reproduce your results. So this is what we want to do. Because the research cycle, as you can see, you will create the data, and you or someone else's process data, but we want you to process, and we want you to do the analysis, and you can do a collaboration too with a bioinformatician, of course, but we want to give you as much power as you can get, and then you can do anything, like many things yourself, and then you're gonna consult the bioinformaticians when you need. But this is a cycle, because when you do this, you analyze the data, you're happy with the results, and then you preserve the data, you share this with your collaborators, and I give the access to your collaborators. But in the future, you decide a new thing, new experiment, and want to compare those results, the old ones. And you need to reuse that data. So this is a cycle. You're gonna see that when, you know, I'm sure many people among you you already know the cycle. So you're getting into this cycle, you create data, process data, analyze data, you go back. And you realize that, oh my God, like I need to add one more sample here or another group here to compare this and that, because you're always thinking. That's why you have that like the power to do these kind of things yourself in your own time. And this requires, of course, some, there's a learning curve there, and some investment, time investment that you need to do to come to that level. But in long term, you're going to see that you're going to turn all the cycle yourself. And if you need anything in the analysis or processing, you're going to work with other people, but you will have the control. This is what we want. Otherwise, projects will not go how you want, and it's going to take forever. So for what is required now, the best practices of creating pipelines or processing data is here. So for every result, again, I told you like how you can keep track, like how you produce the data. Every single command you write, put them into a file, put the versions of the software, put the parameters you use, and be organized. And this is what we started doing using Dolphin Next. I avoid doing many manual data manipulation steps. I create my pipelines. Whatever I do, I put into the pipelines. Because in the future, I know that I'm going to come back and I need to rerun that thing because of this cycle. And if I don't create my own pipelines, I fail or lose that month that I told you. But if I put them and I run those runs in Dolphin Next, that you're going to see that I avoid these manual data manipulation steps, and in the future, I can rerun in 1 minute. So you're going to see it's so easy, those things, when you organize yourself using a tool like we have. And also you need to archive exact version of external programs. This is the things that I was talking about, like Docker images or some containers I was talking about, that we create them for you. If you really into these things, that we can like send you some articles, like some tutorials that you can create your own containers too. Those are very useful. We support them in our cluster. Once you have the container, you can run all bunch of software that you used 10 years ago today. Okay, something like that. Like, even I'm talking about, like, if you create this today, 10 years later, you will still be able to run exactly the same things you did before. And whatever you write yourself as custom scripts that I'm talking about, like, you can create your own scripts using VI, put a couple of commands there, get the input and put the output into a directory, for example. But you need to version them because they never stay like how they are. You improve them. So make sure you have different versions of all the custom scripts you use. We have like huge help here in Dolphin Next to track down all single subversions or revisions of your scripts that you have if you start using Dolphin Next. And in the future here, if you are in a collaboration, for example, Bruce here is in a huge collaboration. He has a huge extensive knowledge about this kind of collaboration with like multiple institutions, and he is managing here. Bruce is here, by the way. You can talk with him. You can learn from him, like how he is managing all these things. It's like very hard job. So you have to, he has this experience if you want to talk. But basically they are doing this, like, they put all the files into the cluster and they are sharing data with people, giving access to them. They are running all these things and they are running this cycle. So, but in Dolphin Next, how we are doing? So Dolphin Next is this one, pipeline management and analysis system. So basically, if you want in the future, you can create your pipelines like this, a drag and drop interface, as you see here, and you define your inputs, outputs. Let me go back again to show, like you can, let me show you. You can define your inputs. These circles are your processes or scripts that you can define your own small scripts, basically just commands inside. If you know how to run those things from command line, you can easily put them here. The only thing you need to do is you need to define what is your input, like FAST2 file, and what is your output, like BAM file, alignment file, for example. You can, for example, in this case, we run star. The inputs are reads, and you get another input if they're paired and or single and reads. So when you get them, you get an alignment file. So then you can run another tool, for example, RCQC, and you can create huge pipelines. But the things we want to do here is we want to create this modularized system that you don't need to focus on the same thing you are doing again and again. For example, star. I'm using star in many different pipelines. One of them is like RNA-seq pipeline, another one single cell RNA-seq pipeline, another one is, I don't know, small RNA-seq pipeline. Like I have lots of pipelines, but I use exactly the same thing, but I did it once and I use it anywhere in many places. For example, FastQC, I did it once. And you don't need to reinvent the wheel and again. You can just use the things that we created, if you like. You can see the code, you can see all these lines, like which command we are using, and the versions and everything here. And then you can put them together and then create your pipeline. But we don't ask you to create these things as band scientists, unless you want to. You have the power, you have this technology, you have all this interface to create them. If you want, you can. But we already created for you. You can just basically press this run button here on top, and you come to this page, run page, that I'm going to show you in detail, by the way. But basically, you choose your single end or paired end, or you choose your genome build here, human, mouse, whatever. You fill all this form and then press run button, and it's not going to be this fast, but basically, it's gonna be completed like this. When you press this report today, hopefully you're gonna start your own run in Dolphin Next. But basically you're gonna run everything here for RNA-seq and you're gonna get all these results. So 90% of my problems are solved here already by just running this RNA-seq pipeline, for example, if you're working on RNA-seq data, because you can do many things here. For example, you can check, oops, what happened? You can check this multi-QC, all the QC results. This is important. First thing you do when you run an RNA-seq data, for example, or ChIP-seq, ATAC-seq, anything, any sequencing, what you do is first you check quality of the reads. And these are, for example, lots of QC checks here. These are alignment qualities. Like how many percent map to genomic regions, how many percent map multi-mappers, how many percent like unique mappers, those kind of things. If there's something wrong, you can see in these plots. Or, excuse me. Or then, oh, I couldn't get used to this, sorry. So then we're gonna show you, after you do these things, in the report section, you're gonna have lots of other files. Oops. You will have other files there, like tables that you can do differential expression analysis, or other tables that we perform lots of other algorithms to analyze your data. One of them is, for example, differential expression analysis that you did manually here, that we're going to show how to do this with R in the next session. But basically, we have this D browser here that you load your file there, choose your conditions, and then press a run button, and you're going to have differential express genes. And these are all interactive plots. You can hover these plots that I'll show you in the next session. But basically, you can create your own heat maps. You can enter the gene set you want to create a heat map. You can do lots of things writing a single line of code in the browser. So DolphinX gives you this kind of environment that you can do your own analysis by yourself. Maybe many of your problems can be solved here. The ones that you cannot solve in these tools, that's the reason you're learning R and RStudio and other tools. This was basically for Dolphin Next. But the whole thing is not, ending here with Dolphin Next, because as I told you before, all the cycle requires lots of people and lots of software that you need to use, from sample collection to data analysis. For example, if you're working in the hospital and you collect lots of metadata from patients, so there's a software called RedCap there they are using. Or you send your sequences to a sequencing facility, they're sequencing your runs, and there are lots of other information coming from there. If there's a problem, so these people need to be in communication, and these people need to actually know who these people are, and all the information needs to be entered into a single place. Otherwise, these people have their own software. These people in sequencing facility have their own systems. And there's like a problem. There's a miscommunication between these people. Sometimes they don't know even who these people are. To ask them, hey, my samples, I have this problem. And this analysis is asking the band scientists, hey, I have this problem. Could you please send an email to your sequencing facility, wherever you sequenced, and resequence them because these files are not good. Something like that. What we want is like, we want to actually integrate everything into a single place using our metadata tracking system. These people, or these people, whoever were working on these projects needs to enter that information into a system that the cycle will turn faster. And this will know, these people will know, everybody will know, whoever contributed the project, and the questions will go to the right person faster if there is something wrong or if they want more sequencing on those samples. So the problems here mostly about the lack of integration, and there is an availability of gap too. That's why there's a bioinformaticians have limited time. There are not many of bioinformaticians around. That's why you need to take the responsibility to in everywhere as a band scientist, because band scientist starts thinking the project, okay? And they're integrating these people in different scale. So you need to have that ultimate responsibility to manage your own project. So you are your own CEO in your lab if you're a postdoc, PhD, or a student that working on, you are your own boss, and you have to manage everything in a right way. And when you have like larger projects, when you're contributing to other projects, again, like, you need to have in that kind of organization to be able to successful. And when you need to scale your projects, DolphinX also allows you to scale this on other computing units. For example, you're doing this project with multiple institutions. They have a cluster, you have cluster. What you can do, you can actually utilize their clusters too. You don't need to open your cluster to other people. In this case, for example, Bruce had the power to create a project and give access to the people in other institutions in our cluster. But you don't, and not everybody has this power to be able to do that, okay? So what you do, basically, you need to utilize their resources in their own clusters if you're doing this, but you need to still collect all that information into a single place. So you can do these kind of things with DolphinX. Because we have all these programs like Dolphin Meta, Dolphin Portal to share the results with outside collaborators, especially, and Dolphin Next and dbrowser and dprofiler for all this operation. And the benefits, they're all integrated. Your efficiency, you're going to see that increase drastically, because the people can create pipelines for you on the fly, whatever you need. And also, it will allow you to like utilize other people's algorithms, other people's effort faster than ever before. So this helped us increasing our efficiency, for example, like at least 10 times, that we can do like lots of projects at the same time with a couple people in the lab. And what we want is actually, like, in the traditional way, the bioinformaticians were doing your analysis and scientists were waiting, they had the ultimate control on data. We just want to change this a little bit. We want to give we want to share the control. I'm a bioinformatician, but I want to share the control with you. And I want you to track down all these cycles yourself. And I want you to ask to the bioinformatician what happened or where it is going within there, because there are like things that you can actually track the status of the samples, what happened, are they analyzed, what are the results, or many of them actually you'll be able to do it yourself. Okay, and this will allow you to outsource some of the things you can do in DolphinX by just telling the bioinformatician in, I don't know, Japan, for example, that, hey, can you please create this algorithm for me? Then you can do the collaboration. They can log in here, create that algorithm for you, and then you can run it. So this is what we want. This is the whole infrastructure, and it's running on cluster, all of these things, and we have this single sign-on system that when you log in everything. And to use all these tools, just talk with us if you need to integrate with these other tools. We are trying to integrate them with like third-party software, like for example, we did with RedCap to get all the data from the cluster, from the hospital. So basically, the meta system that in this automated data processing system, all the fields can be defined by you or like us, people like us, so we can create. But this is very generic forms that we create specific to your project. And basically, these scientists that you collaborate comes here, enter all these, fill the forms, and then you can come here, check all the data that you have. You can update the data, you can collect the data yourself, all about the metadata. These are so important. You can export the Excel sheets, import back, so you can control all the metadata. This is so important, this metadata and the fields you define, whatever you want to use in a downstream analysis. You can define the status of the the data that you want to use in the future. This is crucial because when you want to do, for example, single cell RNA-seq, you need all that information, all the metadata to be able to, for example, it's vitiligo or some other disease coming, it's a lesional site or a non-lesional site. All this data needs to be integrated with the analysis. So if you don't put that information somewhere, that the bioinformatician you want to work with will have no idea what your data is. But then when you put everything here, everybody's job will be easier, and then you're going to track down what's going on your data. And then you can share everything in the standards. So the portal is the tool that we created to share your data with your collaborators. So for example, when you put all that information into your system in the future, even you have 20 samples, you're going to know that you'll see that it's going to increase, and you can integrate them with public data too, and put them here. And then you can run the pipelines automatically. By the way, whatever you put here can call the pipelines in Dolphin Next and run those. For example, this was single cell. When new patient runs comes in, you can enter that information and you can start your runs using this dmeta, and the results will be shared in Dolphin portal with your collaborators if you want to share with everybody. When you publish the data, you're going to see that this is very handy. You can get all that metadata and push the data to NCBI very fast. But these are all connected. All metadata tracking system, your Dolphin Next and Dolphin Next runs and Dolphin Next pipelines and the portal are connected. So whatever you do can affect all of this cycle, and it will allow you to track all that information, all the software you use, all the parameters you use when you're running your Dolphin Next. All the reports are going to be organized, the number of reads, or this is single cell, for example, all the files you need to use to do all this downstream analysis, everything is in your control if you put like this. In this case, for example, the single cell data, and you can go to portal, share the results, put your all this heat maps that you're going to use in your paper and whatever you want here. And we already have these pipelines, but I'm sure when you start working on this, you're going to see that you need to change them. So we allow them, we give them as a template in DNext, and you change them when you press run button, and they're going to be in your portal, shared with your collaborators or yourself for your own records. So then hopefully what we want is basically, like Arthur was working on this project, was usually when you work your own data, even before you start designing your experiments, you want to check public data. And sometimes you want to integrate your data with public data. So there are lots of things you need to do there. You need to collect their metadata. Usually they are on NCBI. And we have like easy way of pulling data from NCBI, processing them using the pipelines you want to process, like those are going to be standard pipelines, and that is defined by you or with the people that you are working with us sometimes. And then you can actually compare. In this case, for example, Arthur like collected data from NCBI, like lots of donors, different projects, even like some healthy data, some disease data, from different type of data to compare vitiligo with other diseases. And he found extremely interesting things there that we couldn't find otherwise, because we don't have this data to compare different disease types. That's why we are trying to simplify all the system that you can compare your data with public data that we did, and we can talk with you. And we started a new project, for example, for single cell ataxic data from brain. We downloaded, there are 3,000 donors there. We started reprocessing data, that data to give access to UMass users now, that hopefully you're gonna be able to use those things if you're working on brain. But we're gonna do this for other things too. So please contact with us if you have that kind of projects. We can start working with you too, that you can have that power to start these analysis yourself. You can have the control. We can assist you. We can help you while you're doing your like this kind of analysis, and prepare all this closed cycle system that you can actually have the ability to share some part in the future within your publication. And basically, in this case, for example, what he did was he used DMeta, Dolphin Next, and integrate different type of data. He used, for example, very fast algorithm Callisto, and he added, Onur added this Callisto module into our existing RNA-seq pipeline, because we thought that Callisto results are very like reproducible with slower pipelines, like Cell Ranger, for example, takes 1 week. With Callisto, it's 6 hours. So almost the results are very comparable. When you look at the correlations of the samples are like almost 1, like sometimes they're more than 95, 0.95 or 0.97 sometimes. So very, very reproducible that when we use Callisto. But you can see the run times. For example, this is bulk RNA-seq data. When you look at these plots here down there, so you can see the alignment speed is like extremely low. You can get your results real fast. You can still run the things in the background with RSAM, but with Callisto, you get the results fast, and then you can start analyzing your data. In this case, for example, he compared lesional and non-lesional, lesional and healthy, and non-lesional and healthy, and find group of genes and cluster them and in different public data and put them together using the browser. So, and he didn't write a single line of code here when he was doing these things. So but of course, to do more operations, you're gonna write code. But basically, doing many operations, you'll see that will be easy, fast, and at least you're gonna get going what you're doing, and then you're gonna talk with other bioinformaticians or with us if you need more. And the last thing that about this part, I think I need to talk that this kind of thing that we want to create in the future, that whatever you do, we have this Dolphin profiler that we created now, that Arthur is working on that too. What we are doing is like basically we are trying to profile bulk data using single cell data, just to find, like, for example, you think that this is a liver cell, but how many percent actually is alpha cells, like beta gamma cells? You can look at your data by the help of single cell data. So those kind of things that we are into now these days, that we want to analyze. Our dream is analyzing all NCBI data, for example, and give you an access to that all process data, that you can find like how pure your data is, for example, that you created, where it is located in a bunch of data. We start with brain and like skin first, and then we're gonna extend this in the future. So please let us know what kind of data types you're working on, and we can prioritize our data sets according to your projects. Then you can start accessing and profile your own data in the future yourself using our tools. So I want to thank all this group, especially Artur and Onur. They are working a lot on these projects, and all the other members, lab members. This was Jake's project in John Harris lab, and Wei is also working a lot on this project. So I think overall what we want to do is this. Now, in the next section, you're going to learn how. So, and if you invest enough time on these things, you're going to see the value, and you're going to see everything will be much easier in long term too in your projects. Okay, let's give like 5 minutes break, and hopefully we're going to go over Dolphin next. You're going to start with doing things here and then start your own run, hopefully today. Okay, just 5 minutes break. I'm gonna come back on like, or maybe 10:55. Let's do 10:55. Any questions? I got one question there. That was good. So in the future, like if you have a bulk data and single cell data, but if your bulk data covers more cells than your single cell data, then you are bound to your single cell data. So you're limited by your single cell data. So if you want to do this deconvolution to find out the percentages of your bulk RNA-seq data, then it is not going to find the cells that you have in your bulk cell, extra cells you have in your bulk cell. So it's how it is. But that's why the reason that we want to put all these things together, like different type of data, different conditions, different cell types, different organisms. Then our dream is in the future, when you come with your own data, you're going to predict what you have and find the best parameters, algorithms to run your data. So this is hopefully going to happen. But Arthur, do you want to tell anything about it? Anything about profiling?
I wish I could, Alper. You actually summarized it beautifully. So,
okay.
So I guess we're good. Yeah.
Okay. Let's continue. And I will share my screen again. Okay. Okay, so I'm gonna do like a kind of demo too. Okay, let me share this. That now you can, maybe all of you guys should have your accounts. So let's start what we're gonna do today. So first, we're going to look at Dolphin Next. This is our graphical user interface for reproducible pipelines. And we created this with Onur and a couple of more people. And what we're going to do, we're going to look what it is, where it is, and how you can log in, and how you can start running pipelines and reports. So in the bootcamp, actually here, what was it? In the session 5. So we have like, all this, how to sign in here, the link should be here. And there are like some explanations about what you're going to do, and you're going to have your homework. So you're going to run one RNA-seq pipeline using public data. You can upload your data too, but I'm going to show a couple of things here. Then I will give you time that you can do yourself too, or you can do when I'm doing too. So basically, Dolphin Next installed here, dolphin.umassmed.edu. But there's one more thing in the future. If you want to install into your environment, into your own laptop, like whole Dolphin Next can be installed there. So in the session 5, we have an optional session here actually for advanced users. So if you are like feeling yourself happy in the first 3 sessions or 4 sessions, that you could do all the homeworks, you could create your own runs, like, you understood cluster, environment, running things. So I suggest you to do this optional session to building your own pipelines in Dolphin Next. So this is like from beginning to the end, you can actually start creating your own pipelines. We have this session, but we are not gonna go over this today or in this bootcamp to creating your own pipelines. We're going to show you how to run them and how to in here, how to run our existing pipelines. We have many different pipelines. Let's go over them. So to be able to see them, you can go here, dolphin.umassmed.edu, and you're going to see we have single cell 10x pipelines. We have in-drop pipelines. Some people are still using in-drop. We have RNA-seq pipelines, star, high set 2, top hat, or we have a whole RNA-seq pipeline. You can choose which aligner you want to use. You don't need to go the specific pipelines. And basically, for example, this is one of the pipeline, how it is looking. So all these things are, again, like, the modules here, like the processes, we call them processes. So when you want to visualize your pipeline, let me show one example, real example. So for example, when you log in to the system, you're going to see like this. This is the main screen. You're going to see some of the pipelines here. So when you want to see what it is in it, you can press learn more button. So when you come inside of that pipeline, you can see all these versions of the software that we are writing. So when you're developing your own pipeline, if you come to that level, after you pass that developer training, then we suggest you to write like all the steps you are doing there, and the container we want you to create, or use our container. We install all the software here. And if you organize like this, it's going to be easy for other people when they come in, they can understand what's going on here. This is very good for visualization. This is revision 6. See, I told you that we have many revisions. So in the first version, like for example, I can go back and still run the first version, revision 0. So this was like very simplified version. So the first time when we created it was like this. It was smaller. But then we put like lots of other things. We improved the pipeline, the modules, but each module here, I went to the back here, each module here is here actually. You can see what it is inside. See, for example, this is check and build module. So you learn how to create your own index files. In this module, we are checking if the index files exist in your environment. If it doesn't, it's going to tell the pipeline that, hey, create these index files for this MM10, or you come up with your own genome. This will do that for you. So we created them like this. So, and then we put like other modules, for example, star module. This is for the alignment. And it's checking and building the indexes themselves too. We put it there too, that you can use this individually without all this RNA-seq pipeline. You can use the module 2 in your own pipeline in the future if you want. You don't need to reinvent the wheel. This is what I was talking about. If you want to see the code, you can come and see code here. See, this is not that difficult. Looks like difficult, but when you learn, for example, make director, you learn how to run the star. We just make it more complicated because we want to support different things. We want to check, for example, if the index file exists, we don't create. We just create a link for it to use existing star index files. Then it's going to be much faster. So you don't need to recreate the index files again and again. In this case, it's just checking. If it's not there, if it is there, it's creating a link. If it is not there, creating the index files that you learn how to. And here, and going to the map star, for example, you learn how to run your star command. In this case, we have some inputs, the index, location of the index file you used in your command, the reads you use in your command, if it is paired or single AND. So basically, you just run this. We just wanted to support paired and single AND too. See, your star command is here. Basically, if you do the developer training, you're going to learn how to use these parameters coming from your inputs. And then the outputs are here, generated here. You learn, for example, how to convert the SEM file coming from your star aligner to BEM file while you do session 4. And then for single AND and paired AND, we put this if clauses there. Those are more advanced, but you don't need to do everything advanced. You can just put them together like, run start, run my paired AND, and then remove the SEM files that they take a lot of space, and then move my BEM file to somewhere else, for example, and then create, for example, if you use some tools FlagStat, it's going to create some statistics about that alignment, and it will tell you like how many % of your reads map to the transcriptome uniquely, or like multi-mappers and everything are going to be here to do your checks, other checks like pre-processing QC. This is going to be like post-processing QC. We call them like this, that you're going to do your QC checks. But again, this can be so simple too. This can be this like hard too. But we recently had this process test section here while creating your own pipelines, that you can actually test your runs by clicking here, and you can choose which environment you want to test, and you can test the script is working in that remote machine or not. So this is beautiful things that I love now developing here, because it just increased my efficiency again more with these kind of things, that you can write your script here and then test. If it is working, then move on with the next step. So, and then, but what I want you to think, if you focus on like creating your own pipelines in the future, you can still put them here, test if it is working on your data. You can define your inputs here too for your tests. See, we have this inputs section here for the test files. And then you can test, and if it is working, use it. And then you can connect the outputs and inputs and create your own. But again, like in this section, we don't want you to focus on creating, but in the future, maybe, you know, if you feel yourself more comfortable after going over a couple of times on this slides and on this bootcamp, then it would be easy for you to create like complex pipelines in the future. This is the section that we put like all these Docker files, like some more like, you know, that container definitions, that if you want to go like more advanced, you can come and see these examples here. So this is open to everybody. You know, everybody can come and see all the code, all the structure as an example. Then in the future, if you want to create yourself, if you need ready to spend day and night, then you can come here and then learn and ask us. We'll be happy to help you. This is what we want to do. By the way, one student did this, like how we are doing here, and he created the pipelines himself. Like I saw, like he created 17 revisions, and he ran this like hundreds of times. And he used the pipeline he created in his pipeline. We didn't do. He was the boss there. We didn't do anything. We just helped him. So I was so happy with that. So Dolphin Next is just putting, like, you can use any language here, by the way, if you want to write your own. You can use any language. It's language agnostic. Like, you know, in this case, for example, in this part, we use Groovy, a language. In this part, we use just Bash. You know, you learn Bash. Just a couple of commands. And you can put a Perl script here. You can put R script here. You can put anything that I'll show you examples later. But it's beautiful. You can write anything. But once you finish, when you connect them, all these inputs and outputs to create a pipeline, then what it is doing, let's go to that part now. Basically, it's creating this is the RNA. See, this person, for example, Ebru, she is using this, one of the old revisions, not the, maybe she used this like 3 years ago, I don't know. She chose to use this. It's not the revision 6, it's revision 3. See, you can still run the old version whenever you want. That's interesting. Okay, so I'm gonna go and show you like once you're running. Let's go back to the presentation. So this is like how it is looking in the whole pipeline side. So there is run button here. When you go to there, to any pipeline, there's a run button, and then you can press the button to run. So I'm gonna do that now. So, but what we have here, for example, our public pipelines, we prepare RNA-seq pipeline. You can do all these genomic alignments with HiSet2, Start, Top Hat, and Callisto. So you can use like one of them, or if you want to compare, like, you know, I saw people were comparing all of them and trying to choose the best one according to their need. You know, you can actually go to a gene, see like how many percent map with HiSet2, Start, or Top Hat, or Callisto. You know, if there's like huge discrepancy, you can choose the right one. And then you can do, using all these results, you can run RSAM or feature counts. Those are also replaceable. RSAM is doing this with an algorithm. So RSAM quantifies the transcripts using a likelihood function. It means that, like, you know, every read, it is calculating a probability to find the origin of that read on the transcriptome. So by also checking the quality information. So it's more sophisticated algorithms using a likelihood function in the background. But feature counts is just counting the number of reads coming from that gene. It's not using any sophisticated algorithm. But when you look at the correlations, usually they are very high. This paper cited like 3,000 times feature counts table algorithm and feature counts software. So many people are using, unless you are working I don't know, I always use RSAM, but it takes like a lot of time to run RSAM, a lot of resources. For example, you can run feature counts in 1 hour, and RSAM takes, for example, the same thing for maybe 1 day or 12 hours, something like that. It takes a lot of time to run RSAM. So that's why, and the differences are not too much, you know, but for some people like me, you know, I run RSAM and I'm happy with RSAM for now, but I started running Callisto. It's much faster and the accuracies are very comparable. So it's up to you according to the project. You know, you can run feature counts first or together, get the results for feature counts, start looking at it, and then in your publication, you know, you can use the RSAM results to create the plots again, because, you know, they are very similar files. Basically, you're going to get a table of the counts, either from RSAM or feature counts. So I am using sometimes both, sometimes just feature counts, sometimes Callisto, sometimes RSAM. Again, like you need to do your differential expression analysis using these count files, and then look if you are losing a lot of DE genes because you use feature counts versus RSAM, or because RSAM did, because of the sophistication, it's just did something wrong. It could be possible too for some specific regions. Then, you know, you might choose feature counts in that case. I'm not telling this is better or worse. I'm just telling that RSAM is using a more sophisticated algorithm. But here, when you run this RNA-seq pipeline, you can do adapter removal, trimming 3' ends, 5' ends. You can do quality filtering. You know, whatever you see in any publication, you can do like 99% of it. Any RNA-seq publication with this pipeline. You just need to choose to fill the like this form and define all these parameters and then run. The good thing is all these parameters will be recorded there. So how? It's like this. For example, I'm going to go to my run that I created, for example, maybe on Amazon, this one. So I run this on Amazon, for example. So it's so easy to run these things on anywhere you go, by the way. In the future, if you go to another institute, another university, you can still use dolphinx.umassmed.edu to connect your university if they allow you to. Or you can install all this tool into your university or into your laptop and start using from laptop to your cluster. So we can help you do these kind of things. And for example, one of the oldest students became professor in UPenn, I guess, and he is using it from here to UPenn. So he just bought a couple of servers. He still uses it from here. And other people in Europe started using like that. Like people are using from everywhere. But whatever you do is not going to stay here with dolphinx. So you can move them with you wherever you go in the future. So in this case, this is the form. So when you press that run button, like on, so learn run button in that pipeline, it's going to come to this page. This is your RNA-seq pipeline. So what I want you to do today is like, you know, you can choose your reads and mate and mouse genome build, because the data that the example data is mouse data. And you can choose like which software you want to use. But I want to point out here, sequential mapping part, that Oli Rando actually gave that idea to us, that that you can map everything you have, all your data to ribosomal RNAs, and then quantify how many percent went to ribosomal RNAs, and then repeat masker, and then add as many sequence sets as you want to quantify. For example, if it is total RNA-seq, you want to quantify, you want to remove first the adapter, that there's a section for that, and then use adapter. After adapter removal, you're going to run ribosomal RNA mappings to remove them, because ribosomal RNAs are everywhere, and even you use polyase selected, that will be like at least 2%, 3% ribosomal RNAs, that you want to remove them because you don't need to carry them with you while mapping the whole genome. You're going to lose time for that, for nothing. Especially some repeat regions you have, you know, you lose time while mapping them to whole genome. That's why we have repeat masker. you map them, remove them, and move forward. So, and then quantify if something is interesting in repeat regions that is like different in your condition versus control, for example. Or if you have a spiking control, you can map them to ERCC. Or you have like snRNAs, snRNAs, piRNAs, tRNAs, or your own custom set. Like if you have a flu genome, flu FASTA file, for example, or flu sequences, flu genes, or COVID genes, you can put them here. The name of the FASTA file you create, put the name of the sequence, and then it will quantify every sequence you put in there. And you can choose to filter those things out or filter in. So in this case, for example, it will filter out repeat masker, and then the rest will go to whole transcriptome or genome. Means like, you saw that pipeline. So when, for example, one of the module finished, in that case, for example, sequential mapping, when it's finished successfully, that sequential mapping it and filtering out every single sequence, and then the next step, the rest of the reads are going to go to the next step. So it is working like that. We set it up like that here. So, but before this, you can use this adapter removal. What basically do is like, if it is total RNA-seq, if it is longer RNAs, like polyselected RNAs, and if your fragment sizes are more than, I don't know, 300 nucleotides, then you don't need to do this. So for example, adapter removal, you just basically write your 3' adapter. Usually it's an Illumina adapter. You can put it there, 3' adapter, and then it will remove it and then do the rest. So trimer, if it is paired end or single end, sometimes, when you look at the QC, you're going to see that some regions in your reads are overall like has a bias, something like, for example, there's all ends in the first nucleotide. You want to remove them, because if you don't remove them, you cannot align them to genome, because there is n, and if you don't allow errors, it's not going to align anything, Or if you have multiple ends, then you have to trim that 5' or 3' end of your reads. So this is how you do it. Just basically write how many nucleotides you want to trim in any ends. If you keep it 0, it's not going to trim that side. But these are optional. So usually I don't do those things. Usually like I was doing these things 7-8 years ago, but today I don't, because the read qualities are pretty good these days. And there are other things, for example, if you want to quantify where they mapped in the transcriptome, like 3' and 5' end introns, 3' exons, or untranslated region, or something like, those kind of things that you can do with RCQC, or intergenic regions, intronic regions that you can quantify and see if anything is weird in your data. And then you choose like which aligner you want to use. I usually use star, it's faster. HiSet to star, very similar, speed-wise. Result-wise, very similar. But I don't know why I usually use star. It's up to you. Try yourself and compare results and see what you like more. But we don't use TopPet anymore. We still have it here just to reproduce all results. But it's so slow, and these are better now, more accurate. And again, I still use RSAM, but slow. And by the way, this is all, oh my God, this is very old version. See, I ran this like a couple years ago, 2019. This is the first revision. That's why I'm seeing like, there are other things here in the new version. So I don't see them here, but that's good. So this means that I can actually duplicate this one, duplicate run. When I duplicate, it will ask me, do I want to use the same run, the same revision with the same parameters, or new revision? So the good thing is, once you fill this form, they're going to stay here forever. And when you duplicate, they're going to come with you, all these parameters you did before. You don't need to go and remember, because sometimes you use a lot
Excuse
of.
me, Ankur.
Yep.
I have a small question. It's like, sorry for interrupting. I just wanted to know, like, when we are filling this form, is it possible for us to enable all the things parallelly, like the star, HiSet, and everything can be run parallelly?
Yes, you can run parallelly. Yeah, but it's going to take more time. You don't need, but again, if you want to compare once, just to get a sense out of it, I did that. Those kind of, because what we do whatever we put here, these kind of approach help us to compare different algorithms. You know, you run all of them together with your own data. By the way, every data is different. You know, like when I tell that maybe star is working on mouse better than I said because of some measures that I think what better is, if I can define. It's very hard to do that. But for example, if your measure is percentage of map reads to unique regions, for example, if star is doing this better percentage wise, then I would choose star. But if you do this on C elegans, if I said 2 is doing that specific measure better because of what you want to do requires uniquely mapped reads, then you might choose HiSat 2. You know what I'm saying? Like,
I got it.
think like a measure to compare the tools to decide which one is better for your data. But I decided a long time ago, and I said that this my thing is star. They're not too different. I use star.
Okay, thank you.
But I'm not telling you you should. You should try your own.
Yeah. It's basically like trial and error, right?
See, and also all the runs you do here is gonna be recorded there too. All the parameters and stuff. All the reports are going to be recorded there too. So basically, you have this log section here. When you finish, for example, in this case, this is real data. I run on the cluster, finished in 4 hours, and I have all the timeline. Like, you know, there were 6 samples here. How much time they took? Like, I have all that information about my run to optimize in the future to reduce the costs. So this use RSAM, by the way, and you can see RSAM took 1 hour. In this case, these were like not small files actually. In this case, so there's this report section here. You can go and check the alignment summary to see how many reads you have. In this case, see total reads 34 million, 16, 19, 22. So I have that many of reads. And I choose ribosomal and repeat masker in this case. See, it just gave me the percentage, 13, 10. See, in this case, some of them 20%. So sometimes you get, see what we're doing here. We just map it here. I mapped the repeat masker and then remove those reads. I started 34 million and then ended up 29 million before sending them to RSAM or star. So if you put other aligners here, you're going to have like 2 more columns each for each aligner. And you're going to see like HiSat 2, you're going to see top hat, you're going to see others. And the percentages, see, I compare, for example, with RSAM. I mapped multi-mappers are like 13% and uniquely aligned reads 52%. This is normal for a kind of regular RNA-seq data. Should be like this, around this percentage, or maybe higher. So for star, we have like higher aligned rates because, as you might recall, that we have some reads coming from intergenic and intronic regions. That's important. So I do both because I go to the genome browser or IGV to check like some intergenic regions if something is interesting going on anywhere, and then go like everywhere and look, zoom out, zoom in, and look what I have there. So these percentages will tell you. If, for example, if some sample, you come here, 25%, 90% mapped to ribosomal RNAs. That's, maybe it didn't work there. I don't know what happened. So, and then you might need to remove that sample in your analysis. You can decide which samples you should remove by looking this kind of alignment rates to detect problematic samples. So dprofiler, the reason that we created dprofiler, that Arthur's tool, to detect these kind of anomalies that will tell you those things automatically and without any cherry picking. It will tell you, hopefully, which samples are not good to use for your analysis. So this is, it's not fully finished and integrated here, but hopefully it's going to be here and working. So the detailed modules you're going to have here about that's like some detailed runs. For example, you put your flu sequence or HIV sequences or COVID, and for each sequence you defined in your FASTA file will be a line here. And then you're going to have the quantification for each sample of that specific sequence you put. And then you can decide, you can even do your, for example, the people are using this for tRNAs and do differential expression analysis for tRNAs. Something is interesting there after they change the perturbation of the cells versus wild type, for example. And there is another section we have here is R Markdown. That this is a template, by the way. We automatically creates a template for you that this is going to be in the next section. What we do is like exactly in the next session that you're going to learn. So basically what we are doing here is we are like reading the file and running a couple of functions and creating these histograms. There are lots of things to learn here about R. So you need to invest a lot of time. You cannot learn it in one day. Okay, so you need to go and spend time, couple weeks to learn the language. This is a kind of language. And there are lots of packages. You load them, like you load the modules in the cluster, you load the packages using library. And then after you load them, you can use the functions in that specific library. In this case, for example, I have this plot library or rcurl library. In this case, for example, I can use this text connection from this function from this library. So there are many functions that we wrote and put into, for example, this file. And you're gonna do these things like in the next session, but eventually you will learn a little bit to change some little parameters here. You can start like that. For example, I define my controls and treatments in my table that you generated in the last session. For example, control replica 1, 2, 3, and experimental replica 1, 2, 3. The order has to be the same for these columns, and then you have the conditions here. And when you do this, you can apply your differential expression analysis yourself. And by changing some of the parameters here, for example, p-adjusted value or log fold change, you can change the plots here yourself. So, or colors, or, you know, the code is here. You just need to play and learn and create this. How you're doing is you can basically, after you run, you're going to have this file as a template. You can change this, anything here. When you press this button, it's going to create a PDF file. When you press this button here, you can make it like full screen. You can work on here. But so before you come next session, please install R&R Studio into your system, that we have some explanations in the bootcamp session 6 that I put it here. So it's going to be here in the session 6. So please, in the bootcamp session 6 here. So R&R Studio. So please install them, R&R Studio, or try to log in to UMass RC 444. Try these things before you come next week and make sure you don't have any problem. To log in here, there are some YouTube videos here, and how you install to your computer is here. And check these things and make sure you are running these things, okay? And otherwise, you're gonna have a lot of issues. You cannot follow the instructions. And okay, so okay. And then next week, after you learn more R, so in the future when you do your own run, you can add your own like plots here. You can add more things to be more organized. You can add as many R Markdown as you want here. That will allow you to, for example, I had another one here. You can just basically save as and create another file. And then when you press this button, this will create this PDF file. And you can download this PDF file here. You can download our Markdown file. The good thing here, when you install our Studio here, our Studio, you can copy and paste all these commands here from the beginning. And these are going to be accessible if you are using a VPN to run all these functions in your R, in our Studio here. It's beautiful. So you can develop, you don't need to use this interface. You can learn from here, copy them to your R Studio, create, and we're going to show you in the next session. So you can create your R script, and then copy these and paste there, and then learn. And this will the way that how we are training people usually, and then you can improve your abilities to create new plots yourself. So the last thing here that we had that I will show you this in detail later, but we integrated this with the browser. Basically, you can come here and I'll show you more details later, but you can filter some of the zero genes and go and do your D analysis by yourself by choosing the conditions and adding as many conditions as you want. Even you can define your covariate. Thanks to Arthur, he added this functionality to the browser. So let us know, like, if you need anything else in the future, we can put into our list and create things for you too. So, but I will talk about this in the next session after you learn how to do DE, and we're gonna visit this part and how to run the browser in your own computer. Basically, once you run this pipeline, that I'll give you time now, basically you're going to have a table like this. In that table, you have the genes, just transcript names. Usually we don't use this. And we have 4 files here, 4 tables. One of them is for genes, expected counts. So when you see that it's row counts, you can think that row counts, because when you do your differential expression analysis, the counts should be row counts, unnormalized counts, okay? If they're normalized, the tests, the statistical tests, by the way, Bruce is also a statistician, so he knows better these things. So the tests behind differential expression analysis are using a distribution called negative binomial. So it requires row counts. So this is important. I just want to underline that, because I see some publications, like when I review papers, they are using normalized counts and do DE. So you cannot use, for example, this TPM counts and do DE, differential expression analysis, okay? So this is kind of very important thing. When you submit your paper, make sure you did this differential expression analysis with the right file, like this row counts. Expected counts, the name is weird, but this is how RSAM produces the file. But those are like, row counts. You can take it like that. TPMs are transcript per million. So those are normalized per million. So why we do that? Usually we do DE analysis with these files, but when we do the visualization in the browser, we use normalized counts. So again, first do your tests to find which genes are going to be like kind of significantly different. For example, when you compare 2 different conditions, use your row counts. And when you create your like heat maps, use the normalized counts. But we do this normalization after we do the test using that tool. But if you really need to normalize before DE, there are other methods that you can still keep the distribution as row counts, but you can normalize. So, but those are not TPM or RPKM or FPKM. So if you need to do those things, please let us know. Usually we use batch effect correction, for example, if we require this kind of normalization, and we try to keep the distribution to fit the negative binomial distribution. Otherwise, it's not going to be count value, and it's not going to work how it's supposed to work. Okay, so these are the things. And the last thing I want to show you, when you're running the pipeline, so I want you to run now, and then we're going to follow up later. But what I want to show you here is how you're going to run it. So basically, you're going to come here. I'm going to go back on how I came here. I will show you again. So basically, you just go here. In your main page after you log in, you go to. There are many ways to go, but I'm going to show you the easiest way. So you go click Learn More, and then when you press Run button here, choose the version you want to run. Usually I use the latest version, but in the future you might want to run in one of the old versions for reproducibility purposes. But you can create a new run here. So I'm going to create a new run. So for example, I'm going to create a new project first. my test project. And then you save changes. And you select the test project and select the project and then write a run name. For example, my first one, whatever you want to write. And then this will bring you this page. And this command, this button will be waiting. Okay, it's waiting means that you need to fill the form. It's not filled yet. In your case, you have cluster here. Choose cluster. If you define other environments like Amazon, Google, other things in the future, you can run them there too. I choose my cluster. So for the working directory, you need to define where you want to run this, because in the background, you can go and access those files in the cluster. In this case, for example, I want to run this on my like W biocore on my own directory. Could you correct? If that directory doesn't exist, this will create for you, so you don't need to create. Okay, just don't think, just write. When you organize your files, what I do usually, for example, if I do an initial run just for FastQC, I just create like kind of FastQC run, or you can write a name. Again, like my first one. But in this case, please don't use like spaces here, because this is Linux. Linux doesn't like spaces. You can still use it, but it will be so hard for you to use those files in the future from the web or from somewhere else. Don't use space. Don't use any characters like dollars, like all these alphanumeric characters. Just use letters, numbers, and underscores, okay? That's all. Even like I don't suggest using dashes, because R doesn't like dashes. When you load the file with dash or some columns with dash, it changes with dots. And in the long term, it's gonna be hard for you to maintain or work on those columns. It's up to you. Some people are still using dash, even I told them, but you know, it's gonna be harder if you use R. In Python, there's no such problem. If you don't use R, if you just want to use Python, that's totally fine. So the next thing you want to do is here, this should come filled, but I don't know if it's not, just fill it. There are explanations there. So basically, you can write this image to be able to run this, but this should be, when you choose this cluster, this should come automatically. Usually you don't change that, but in the future, if you create your own images like this image means a machine, that all the software is installed in that machine. But this machine is like a imaginary machine, you know, it's not a real machine. It's just working on a Linux machine. It's called container. And we just run this image to create a container. That container includes all that software we need. And these are the things that the folders or storage you want to use. In this case, I want to use like share folder, and I use bind command. So, and the project sometimes, and I just put them 3. Like there are not other things. Your home folder is going to be already mounted in this image, because what it is doing basically, it is mounting this NL storage inside of this container, and all these files that you define will be accessible inside of that machine, that virtual machine. Okay, you can say the virtual machine too, but it's very lightweight. It's already there. It takes a second to run it. If you run it the first time in another environment, download takes like a couple minutes. If it is big, it takes more time, but it's going to come there and then run it anywhere you want. So that's the good thing. Like you can run this pipeline anywhere you want, if that Singularity or Docker installed on that computer. Okay, so, and the second thing you're going to do is like you're going to define your reads. So there's this enter file section. And basically you can add files by pressing this button. There are some videos there to add the files and stuff. So basically, if they're in the cluster, like this, for example, you can just press this button and it will connect to the cluster using your username and bring all those files there. See, all these files came if it is in the cluster. In your example, you're gonna enter some geo numbers to bring, these files are gonna come from NCBI. So if you want to analyze public data, you can bring them here and define a collection name. So for example, in this case, you're gonna bring the files from Vernier et al. Like publication, that there are more information in the bootcamp, but basically you're gonna just enter that numbers here and then press the button to create a file. But I'm gonna just show you like how I'm gonna do this, the ones in the cluster. So by the way, this is here. You can run them, the small files. These are gonna be small files, test files, but you can run them faster if you want to by using copying, pasting this folder. By the way, this supports the files on Amazon S3 or Google Cloud S3 if you have buckets there. Directly, you can download them by just entering that information. Or if they're on the internet, you can use that in like URL to access the files or the files in your cluster. So how it does this know your cluster is because you define your environment in the previous form that will connect to that specific environment, and this folder has to be there. It's there in our case. So our files are fast to initiate the run. And the collection, so I press this button to connect. So when I connect, I get a list of those files. By the way, there's this, like you can move inside of these directories by pressing this folders, and it's not perfect, but it is working. So for example, in this case, our the folder was gzip, gz, where is that? Here, for example, if you double click this, it's gonna go in to that folder. You can see files. So this is FASTQ, and the collection type are paired end. And then when you do this paired end, basically you need to define a pattern to define R1, the first pair. Usually it's like this, by the way. You don't need to do anything. But in this case, I changed the files just to show you this. I put dots into first end, like one.gzip. I put on the left side. This is the R1 pattern, and R2 pattern will be .2.gzip. Okay, so now I can do this. I can add the files, like I can select the file and add selected files. For example, it will add these files as a pair from this directory, and you can give a name, control replica 1. If you want to change the name, change the name. Give a shorter name. Again, don't use spaces, don't use dashes, like anything. Start your samples with the with a string character, not numbers, because when you put a number here, it works, but at the end, R doesn't like numbers. It's just putting X. When you load the files in R, if the sample name has like a number in the column name, in the field name or sample name, so it's just putting an X in front of it, and then it becomes harder. So what I do is like, if I want to put number, I put like sample1 or S1, S2, is like you can just give like this. This will make your job easier in the long run. So you can add one by one, or if you have multiple files coming from the same technical replica or same replica, sometimes sequencing files are like this. They split into multiple files to like make copying the files easier. So what you can do, like for example, you can select multiple files, and then like for example, I'm gonna select these two, and the third one with command. You can use the command, and then it's selected files. Oops, sorry, I did wrong. You need to merge selected files. So this is going to merge the selected files and give one name. So you can do that too. So for example, I put the controls. In this case, I needed to do that, for example, and you can do this. And I put this too for experiments, for example. So in this case, don't do like this, by the way, in your case, but unless you really want to merge the files physically, so they're going to be merged, and you can have two samples in the downstream analysis. But basically what I do here, I want to add all. And whatever you write here as a pattern will be removed while naming, but you can always change the name. And then you give a name, for example, tests 2022 bootcamp. So I just give this name. And if you want to get a backup to another directory after merging, like after changing the file names, whatever, you can write the folder here. Or if you have an Amazon connection that up there that you define in your profile, then you can write S3 bucket that it will get a backup there. I always, most of the time I use that. And Google storage, you can have an account on Google, and then you can, these are optional, you don't need to, right? But this is required. So after save the files, they're gonna come here. You can add more files. You can choose other collections you used before here. But basically, you're just gonna click here to add more files. You can add one by one. You can remove some files if you want, like this. And then when you save, see test bootcamp. If you want to go back and change anything, come here. Sometimes, you know, you move files from one location to another, change the location manually and save. Okay, so it's how it is. And so let's start. Now, the rest is like basically you're going to choose the human genome. Like here, if you want to finish this fast, just use the test, mouse test MM10 here. But in your case, in your homework, you're going to use the real files. Don't use MM10 test, use the real MM10. Like one of the, where is that here? Gene code, for example, use this or this. So gene code, it has more genes than RefSec. RefSec, all the gene annotations here are as a publication. So if you want like more stringent definitions of genes, you use RefSec here. Less stringent definitions, new versions of annotations, use GenCode data. For ensemble, different annotations that we have here for different things. If you require any other assembly or any other like annotation, just let us know, we add them. Or you have custom, you can add yourself and use it in your own directories if it is something custom to you. For example, custom means sometimes I want to map everything together with 18, 19 and mouse together. It's a humanized mouse. The data is coming together. And then you cannot map them separately because you cannot, because there are some read similarities between human and mouse. So you want to map them together, put them together. And then what you do, basically you cut those two genome files and then put the location into here, custom. And then it will the custom. And then you need to fill up these GTF file location for that specific genome location. If you have common DB, you need to define all these things. But if you don't, as you can see, when I change this to custom to MM10, for example, gene code, all these filled by automatically. You don't need to do anything. Okay. So these are our shared locations, and you can see what they are. You can go in the background. Hey, I want to look at that GTF file I used in my publication in the future. You can go and see what this file was. And some people will ask you, hey, could you please send your annotation file and your genome file? And then we put them onto internet too, like in a web accessible area, they can access, or you can talk with us that we can, if it is not there, we can share with your collaborators to all these files, or you can share if you know how to, like FTP to somewhere else, basically. Okay, so I think what you need is that, and you have 10 minutes. We're going to be here before we close, but I want you to log in here and start doing those things there. And if you have any questions, if you're stuck anywhere, you can ask now. I'm going to wait here, and we're going to wait here until 12. And then you're going to do your homework later when you have time in the weekend, whatever. But try to finish them before you come in the next session. if you don't finish, we already prepared those files for you. You can still do that. But if you finish, you'll know exactly where you are, where the files are. So basically, when you finish this, like wherever you use the directory, I want you to go there and look. It's not there now, but my run will be here. And the files, reports, everything physically be there. You can remove some of the intermediate files you're not going to use, because you're going to see like keeping your original files and rerunning these will be much easier. Everything to create intermediate files, BAM files, and everything in the future, because you're going to see this run will finish in 4 hours, for example. But again, like space is so expensive, so make sure that you don't keep all these intermediate files, remove them, but run when you need it. Again, you can always rerun these things. Come here, and then so when after I run this, that'll be a rerun. Actually, I can show you. I'm going to go to one of my run again. This is another pipeline, but I'm going to go there. See, completed run. You can always rerun it. See, if you need the intermediate files in that location, even this file is not there. As soon as your original files are in that directory, this will run. Or the good thing here, again, I'll show you, you can duplicate the run. See, if you want to run these things and keep the old run, you can do that. You can duplicate and then change the name. You can change the name, by the way. Come and click here, you can change the name. And if you want to go to pipeline, you can just press this button to go to pipeline. You can change or the project to see what other runs you have in the project. You can press here or here. Okay, overall, I think what we have is this to how to run the runs, and then we will continue next week the rest. But again, please try and ask your questions if you have any. So the question again was about should I use a project directory or bootcamp folder or any other places? You can use any storage you have. I was using my nearline space. You can use your project space. Usually with the real files you're going to download from NCBI, actually, you need to use your project space. Otherwise, your home will not be enough to finalize the run. Or we put like an explanation there into Bootcamp that you can only download 1 or 2 files. It's going to be smaller. Dropbox. Have you tried with Dropbox, Onur? If you share,
right now it doesn't supporting. Maybe there are ways, but we haven't integrated.
Okay, yeah, we haven't directly integrated with Dropbox. You need to download it to your computer and upload them to the cluster. Usually we don't use Dropbox to put FASTQ files because it's not for it, because we have limited space in Dropbox, but we have unlimited space in the Amazon. So canine, do we have dog? Onur, do we have dog here? I think I worked on dog once, but the problem about dog was about the annotation. So it's not well annotated and also well assembled. So assembly wasn't good in dog. Do we have anywhere?
I'm not sure, Alper. I need to check.
Yeah, we can add it. We can add one version.
Okay.
But again, like not the world is not perfect in the assembly world because, for example, I was working on bear. There was no assembled bear genome that time. I used dog genome, then bear, and then Jackson Lab, Ron, we are collaborating now. did the assembly, first assembly and first annotation, and started using there. Yeah, you can use WinSCP2 or FileZilla. Yeah, there are many FTP clients. You can use FileZilla, you can use WinSCP most likely in Mac too. I suggest I use FileZilla, but there are many. If you just search FTP client, you can use it. But more or less, you need only your username, password, and the host. And that tool needs to have SSL support. That's all. And then you can connect. There's a little learning curve here. You learn a lot of things. I talked a lot. Please ask anything is not clear. There are little
Akbar,
things.
I have one question.
Yes, please.
In case if we have to, build our own pipeline, not now, I'm just learning, like in future, after getting to know all these things, in case if we have to write our own, build our own pipeline, in that case, from listening to your lecture for this past 2 hours, I think we need to know, correct me if I'm wrong, we need to know R script, and I think we need to know the basics of all these passkey files and SAM files and creating directories and all this stuff.
Actually, no, but like, look, whatever language you know, even you don't need to know language. So if you know Bash,
all right,
for you to create some pipelines that you want to process your data. But again, if you know R, you can do things easier. If you need to take, like these pipelines are so generic, this is a platform, so you can create anything. It's up to you how you want to.
Yeah, I think my question, I should reframe it. My question is like, in case if I have to build a pipeline on my own, what do you suggest? What are the main prerequisites I should be prepared before building that? So that would be my question.
By the way, whatever you need is in that tutorial. So just follow the tutorial, all the prerequisites, everything is written there. But basically, we are not requiring knowing any language. The only thing you need to know is like Bash, the first session. You need to understand that first. That will make your job easier. The second, you don't need to understand too much about the job submissions and stuff. It's doing automatically for you. It's using the notes, and you don't need to know that. But if you know like R a little bit to tweak some templates that we create for you, that will help you. But again, like, you can, if some of you guys know Python, for example, and reject learning R, that's fine too. You can write your program with Python or Perl, any languages. This is how we created these examples that are our usage, but it doesn't have to be like that. It's up to you. You can use just Bash, for example.
Okay, thank you. Yeah.
So why we are using the command line version of the pipeline sometimes is because some people don't like using a web interface. They like command lines, especially old bioinformaticians like me. We like command lines, but after DolphinX, I switched my site. But I see there are still people, they like using terminal. They feel themselves like bioinformatician. So it's up to people. I like using the interface now. And if I need terminal, I use terminal, but usually I do everything on DolphinX. Even like I can show you one thing there that I was working, and I love doing it now because Onur added that new functionality that we have. So I started writing this new Perl script to calculate like this, for example, this equation here in this paper. Look, like this, there are some, these are count values, for example. I want to calculate this division and log to A, and then F values, and then I will calculate S values to create the heat map above here. See, I want to create this heat map using those equations. So what I started doing, I wrote this, count A, the A values, by the way. For example, I can show you the code. It takes time, but this is a very complicated calculation, but I'm using like each barcode and like, I wrote the code here and I tested, it's working beautifully. These were my inputs and I did everything here. And my resulting for A files are here prepared and R files. And now I'm writing a new one to divide those R values and then get log2. I did that, by the way, sorry, here. But I started creating calculation of the other two equation, F and S, and I started doing it here. Oops. I don't know why it's not opening now. Maybe it's too small. Just zoomed in. Oh no, what is wrong with this? It takes time now. Maybe my computer is dead.
I'm not sure, but maybe you can refresh the page.
I don't know. It should come. Yeah, good idea. Maybe I came too fast or something. I don't know. Oh, it's not there. Interesting. Maybe I didn't save it. Okay, I'll put it back. And I put this connection. Interesting. It's not coming. Whatever. So it's coming out. Something interesting is happening now. I'll look at it. But yeah, so you can start writing your own code like how I showed and then test it. And if it's working, I'm going to go to the next section to create the plots. It's very handy, these kind of things. Okay, I think we are done today. Thank you very much for listening. Talk to you on Thursday. Bye.
Thank you.
You're welcome. Bye.